Muscle excitation contraction coupling is just a fancy and technical term for muscle flexing. This process has two parts. We will begin by first labeling and understanding the parts of a neuromuscular junction. This is where the neuron connects to the muscle cell. The somatic motor neuron sends signals from the nervous system. The synaptic bulb is the connecting link between the two. The synaptic bulb is filled with vesicles containing acetylcholine. Acetylcholine receptors are located in the folds of the motor end plate. The motor end plate is on the sarcolemma, or the membrane, of the muscle cell. The transverse tubule, also known as the T-tubule, is the vertical tube located on the cell. The DHP receptor is located in the T-tubule. The rhyodine receptor is located in the sarcoplasmic reticulum. In the sarcoplasmic reticulum, there are lots and lots of calcium molecules. As a nerve impulse travels down the somatic motor neuron into the axon terminal, it pushes vesicles containing acetylcholine, down to the end of the synaptic bulb. Once they are released, the vesicles travel down and attach to the acetylcholine receptors. This triggers an action potential. Normally, the charge on the outside of the muscle cell is positive and the inside charge is negative. When the action potential is released, the cell is temporarily depolarized, causing a switch in charges. The outside becomes negative and the inside becomes positive. This action potential travels down the T-tubule and causes a reaction with the DHP receptor. The DHP receptor opens the rhyodine receptor, which is actually more like a gate, releasing many needed calcium molecules. The calcium molecules will be used in the next section. The thick filaments in muscle fibers are called myosin, and the thin filaments are called actin. In a sarcomere, actin and myosin are alternated, giving muscles their striated appearances. When calcium is released from the sarcomatic reticulum, they attach to tropin. Once the calcium and tropin are attached, it causes a movement of tropomyosin, which then exposes the binding sites on actin. The muscle contraction truly begins when a bound ATP molecule that is attached to the myosin head is hydrolyzed into ADP and inorganic phosphate. This causes the myosin head to outstretch and attach itself onto the now exposed binding sites on actin, forming a cross bridge. A power stroke then is exerted, causing the actin filaments to be shortened, which then flexes the muscle.